Hello, this is Mr. Merrier with Noteworthy Science. Today we're going to be doing the higher level subtopic B3.3 Muscles and Motility. Under the curriculum matrix, this falls under theme B, form and function, and under the level of organization of 3, organisms. Some guiding questions to keep in the back of your mind as you're learning this topic is, how do muscles actually contract, and how does that relate to movement? How does that cause movement? And another one that to keep in the back of your mind as we're learning this is, really, what is the benefit of animals even having muscle tissue in the first place? As mentioned, this is a higher level only subtopic, therefore there are no standard level learning outcomes. In higher level, there are 10 different learning outcomes that we're going to explore today. Let's get started. Before we get into how muscles actually work, I think a great place to start is, what is movement and what are the different ways that movement looks like across the different organisms of the planet? So recall back in A2.2, we looked at the 8 key features of what it means really to be alive, and features that all living organisms can do. And recall that one of those features was movement. That means everything from bacteria to plants to fungi to animals, all of these organisms move. So how does movement differ between these organisms? And that's what we're going to explore first. So movement is really variable across different organisms, and 2 broad categories in which we can explore this is an organism sessile or is an organism motile? So let's explore that first one. If an organism is sessile, then what that means is that as an organism, it doesn't move its body from place to place. Typically, as an adult, wherever it's found, it remains fixed in that position the entire time. If you're wondering how a sessile population can move from place to place, well, it's typically their juvenile stages or perhaps even the gametes are what allow the population to spread from area to area. But typically, as an adult, sessile organisms remain in a fixed position for the majority of their life. But that doesn't mean that they don't display movement. So this first learning outcome here is exploring what is a sessile organism, and students should use at least one example to help explore this. I'm going to be exploring 3 different examples just to give you some more variety. But keep in mind that none of these are required examples. You can choose whichever example you wish. Let's start with one of the coolest plant examples that I can think about, and that being a Venus flytrap. Like all other plants, these are sessile in that they are incapable of self-locomotion, i.e. a plant cannot bring its entire body from one place to another place, typically. So what we're discussing here is, yes, the Venus flytrap is sessile, but it doesn't mean that it doesn't display movement. And the reason I chose this as an example is the way in which it displays movement is fascinating. So the Venus flytrap has these specialized leaves that actually have these hairs that act as triggers. And so that leaf is going to ooze some sugary, nice smelling, fragrant nectar, and that's going to attract insects to come and feed on it. As the insects come and feed on it, they're going to accidentally trip some of those hairs. And so when that first hair is tripped, there's almost like a timer. And if a second hair is tripped within around 20 seconds, then there's a very fast closing motion of the leaves, and they interlock together, trapping the insect inside. And then what happens next is that they secrete digestive enzymes, and the plant is able to absorb the nutrients from that insect. And you may be asking yourself, why does this plant do this, but most other plants do not? The big reason for this is that this is actually an evolutionary adaptation, in that these plants typically grow in nitrogen poor or nitrogen deficient soils. And as we know, nitrogen is an incredibly important nutrient for plant growth. It's involved in protein synthesis, it's involved in nucleic acid synthesis, etc. So usually, if a plant is growing in an area with very poor nitrogen, it's not going to fare very well. So as an adaptation, what these plants are doing is they are supplementing that nitrogen by catching insects and using the proteins and nucleic acids that they're absorbing from the insects in order to supplement that. The next example is that of a sessile animal, and this animal is one that we'll probably be discussing several times in this course because it's a key role ecologically, and that is the staghorn coral. Staghorn because the shape of its exoskeleton and the protrusions kind of look like a staghorn antler. But this is an actual animal, and I think a lot of people forget that corals are animals, but they are sessile animals. So as a juvenile stage, the larvae and the polyps, they can actually be carried through ocean currents, and then the polyps are going to aggregate in a colony, and they're going to set themselves down on a piece of rock, and that's where they're going to spend most of their lives. Those colonies of polyps are then going to be surrounded by an exoskeleton that they create, made up of calcium carbonate, and that's that rocky-like texture that you see from corals. It's their exoskeleton. So they are sessile, and that once it's in its adult form, it's going to stay there, but it does display movement. And one of the ways that it displays movement are through its tentacles. So those tentacles have these stinging cells called nematocytes that are actually able to sting prey, such as like very small crustaceans or plankton, and that is then able to be digested by the actual coral. Now, it should be noted that this is not the primary way in which they obtain nutrients. Actually, the coolest way that they obtain nutrients, and most of their nutrients, is through a symbiotic relationship that they have with an algae, that algae being Zooxanthellae. So this Zooxanthellae algae is actually photosynthetic, and they are producers. And so there's this really cool mutualistic relationship where the algae are getting the benefit of having a strong, stable habitat, and the actual corals themselves are getting the benefit of the photosynthetic products that the algae is making. So because of this relationship, this is what gives them their bright colors. This is what makes them such keystone species in aquatic environments. And the last animal example that I want to use is one that's kind of close to home for me in Canada, and that is unfortunately the zebra mussel. This is one of the most devastating invasive species in the Great Lakes. And what a zebra mussel is they're a freshwater sessile animal, and they effectively set themselves on a rocky outcrop, and they tie themselves there using these things called byssal threads. They're almost like anchoring points. And once they anchor to a substrate, whether it's a rock or something else, they then act as filter feeders. So water is going to pass in through these siphons, and they're able to extract plankton or nutrients that are floating through the water. They're a good example of a sessile organism because as an adult form, they don't displace themselves from place to place, but they can still display movement in that these byssal threads can actually be protruded out, and they can actually anchor to other points. In fact, this is what made them so devastating to the native species of mussels, because they were actually anchoring on top of native mussels, and so they were basically sniping all the nutrients before the natives got to it. Now, I think when we think about movement, we probably are thinking of a motile organism. And a motile organism is one that is able to do locomotion, meaning it is able to move itself from one place to another place. So the same learning outcome asks students to be able to describe motile organisms, again, using an example of their choice. I'm going to use 3 of my favorite animal examples to help demonstrate different ways that motility looks like in the animal kingdom. The first one is one of my favorite animals of all time, and that being the cheetah. So the cheetah goes without saying, is an extremely motile animal. In fact, it's the fastest running land animal. So it is incredibly fast. I mean, I could talk to you all day about how amazing the cheetah is. It can run from 0 to 100 in 3 seconds. It can reach speeds of over 100 kilometers an hour. It is remarkable. But what adaptations does it have that allow it to do this kind of running? Well, for one, it has an incredibly flexible spine that allow it to have huge, long, flexible strides, and that allows it to gain enormous speed very, very quickly. It's also one of the only big cats that do not have retractable claws. The claws are always out. So it's almost like they're wearing like track spikes all the time for extra traction and extra grip. In order to facilitate their very fast movement, they actually have a very small head, and that imparts a more aerodynamic stream. They have a really large heart, lungs, liver, and that is for the really high metabolic demand that they have during these sprints. And for those of you that are runners, you may be familiar with the 2 major classes of skeletal muscles, that being fast twitch and slow twitch muscles. And slow twitch muscles are those that you would see in long distance runners, whereas fast twitch muscles are those that you'd see more prevalent in sprinters. Well, it goes without saying that the cheetah has a very high proportion of fast twitch muscles. So these animals are really built for lightweight, short burst sprints. And they're extremely well adapted to this. Now I want to discuss my favorite bird of all time, and that being the ruby-throated hummingbird. Hummingbirds, again, I can talk all day about how awesome they are, but they're basically bird helicopters. They're the only birds that are capable of hovering, flying backwards. They have enormous amount of maneuverability, and there's a few adaptations that allow them to do that. For one, they're very, very, very lightweight. Some of them being under 3 grams, and so they have very little weight that they need in order to keep themselves up. Their pectoral muscles right here are quite strong, and they have very flexible shoulder joints. And in fact, if you look at a slow-mo of an actual hummingbird wing, we can't perceive it because these wings are flapping at over 60 times a second, and they're actually flapping in a figure-8 pattern. Almost like if you know how to tread water and you're doing this, this is kind of what their wings are doing, and that allows them this massive amount of maneuverability. Now, if you've noticed, their feet are really, really small. In fact, they're so small that hummingbirds can't really walk. They can only perch themselves and maybe scoot over, but they can't really walk in the classical sense. And that is because this allows them to be really aerodynamic, again, reducing drag. They don't really need to be able to walk when they can fly this well. Something that you may not have known is that these birds are also migratory birds, and they can complete an 800-kilometer flight from Canada to Mexico. And they're able to do this by putting on some fat, something that we've talked about in B1.1. And this allows them this long-term lightweight energy storage. But typically speaking, they spend most of their time feeding on sugar, where they could consume more than their body weight every day just to sustain their really, really high metabolic demand. And the last example I want to talk about here is like such a cool animal. And again, if someone's never seen a jellyfish before, I think they'd probably swear that they're almost alien in appearance because they look so different from so many other animals. So the Australian box jelly, you probably know it as one of the most venomous animals on the planet. And that's true, their venom is incredibly potent. But what I want to talk to you today in this context is why there's such an awesome example of a motile organism. So you might be wondering, don't just jellyfish just float from place to place? Well, they can. This structure here called the bell, they can actually open up their bell and almost act like a parachute, allowing them to drift in currents. So they can do this. But the box jelly is actually a predatory jellyfish that actually can chase prey. So they're not going to chase very well by just floating. They have this extra adaptation where they have this muscular ring at the bottom here called the valerium. And what they can do is they can squeeze that valerium into a tight hole, and they can contract and push water out of this, almost like a jet. And this allows them to move very, very quickly, over 2 meters a second. And so their ability to not only float when they need to, but also do rapid bursts of propulsion is really, really kind of amazing. So overall, this first part is you should be familiar with the difference between a sessile organism and a motile organism, and you should have at least one example of each that you could use to explore this concept on, let's say, an assessment. This next learning outcome is, I think, really, really interesting, and that is the question of why. Why have locomotion? There's, as we've already discussed, there are plenty of examples of organisms that are non-motile or sessile. So why locomotion? There must be a reason for this. And there's in fact 4 major reasons that we can talk about why locomotion has evolved. And these 4 reasons are looking for food, looking for a mate, escaping from predators, and migration. You should be able not only to explain each of these reasons, but you should be able to explain it using one example of your choice. So like before, all the examples that I'm going to be discussing are not named required examples, but I'm giving you a lot of different options that you could use to help explain each of these reasons. So the first reason that we're going to talk about, I think, is fairly intuitive, and that is looking for food. So if you're a heterotrophic organism, being able to move your body from one place to another place is very advantageous in trying to acquire nutrition. The first example that I want to use is one of my favorite animals of all time, and that being a great white shark. These are top tier predators in their respective food chains, they have so many adaptations that I could really speak all day about how amazing they are. So what makes them such good predators and such efficient predators? Well, there's a lot of different adaptations that they have. Probably really well-known ones are they have an incredibly sensitive sense of smell where they can actually detect parts of blood in very small quantities, and this allows them to identify an injured prey far away that they can actually be able to feed on. Another one that you may not have known as is that they have something called a lateral line system. And this travels throughout their body, and they have these little organs called ampullae of Lorenzini, and what these do is that they can actually detect electrical signals being transmitted by muscle movement of prey. So they can actually detect muscle movement from an electrical perspective. Like, it's like really, really wild. It's kind of amazing. From a visual standpoint, they have something called counter shading, which means that the top half of their body is actually dark, but the bottom half of their body is actually quite white. And what this allows them to do, if you're a fish looking up, then the bottom half of their body will actually blend with the bright sun. But if you're looking down, then their dark part of the body will blend in with the dark ocean floor. So they're actually, this kind of shading here is actually for camouflage, really. And another thing that they have that's actually very unlike a lot of fish is that they maintain a really high body temperature. And that body temperature actually allows them to be more efficient in terms of swimming, and actually allows them to hit higher speeds than otherwise would be for an ectothermic organism. And finally, I can't talk about sharks without talking about their amazing teeth. They have these large serrated teeth that are really good at tearing into prey, and they effectively have an unlimited source. So even if they lose a teeth in a particular bout, they have many, many, many rows that are going to consistently, almost like a conveyor belt, replace their teeth throughout their lifetime. The next example I want to use is probably one of the most ecologically important organisms ever that you may not have considered very much, and that is the European honeybee. Honeybees are enormously important pollinators. In fact, over 60% of all the crops that we consume are a direct result of animal pollinators like the bee. So what exactly are they doing? Well, they're flying from one flower to another flower, and they're not pollinating consciously. What they're doing is that they're looking for nectar. So they're looking for the sugary nectar that they can then bring back to the hive. And they're really, really great flyers. They have 2 sets of wings, and they're able to beat their wings at over 200 times a second. And like the hummingbird, in fact, even better than the hummingbird, they have enormous motility. They can move in pretty much any direction that they choose, and they can hover, allowing them to enter really small spaces like within flowers. And they can move from place to place really, really easily. Their wings also have a hydrophobic coating, and this allows them to fly fairly efficiently through fog and dew without the water actually saturating their wings and making them heavier. Furthermore, their eyes are they have these amazing compound eyes that allow them actually to have fairly good vision, and on top of that, they are sensitive into the UV range. So unlike us, they can see into the UV, and this allows them to detect certain flowers that actually have markings that are only visible in the UV. So it allows them to very accurately pinpoint a specific kind of flower that allows them to go there. The next example of why would an organism be motile would be, well, looking for a mate. And this is a reality for many sexually reproducing organisms. I would be remiss if I didn't use the Western Paradia as an example here. So this is one of the species, this is one of many species of birds of paradise. The males have an amazingly incredible adaptation in their plumage and in the very ritualistic dance that they do in order to attract females. So the way in which their sexual behavior works is that the females will assess males based off of what's called a courtship ritual. So the males are going to clear an entire area out in the forest floor, and then once a female comes to perch to basically observe, the males will engage in a really specific ritualistic dance, and that includes like shaking their head back and forth, bowing, extending their plumes almost like a ballerina skirt. They have an iridescent patch right here that they'll flash to the female and show that. And what the females are doing is they're effectively assessing the quality of the males through this ritualistic dance. And if it's acceptable, they will then be able to engage in sexual reproduction. And so these ornaments and these dances are really all to do with motility. Really, all of this exists solely to attract a female in order to successfully copulate with her. And the other example I want to use here is one that you may not be as familiar with. I think everyone here knows a lion, and everyone here is familiar and probably thinks, oh, the male lion is like, you're the king, you're the best. But that's only if you're successful. Actually, the life of a male lion is kind of sad, to be honest. Once a male cub is born, they're going to be with their pride for a little bit, but the moment that they reach essentially adolescence, they're kicked out of their pride. And that's because a pride has a single male with a bunch of females, and any other males are going to be seen as a competition. So they get kicked out of their pride, and now the males are basically going to be on their own, or potentially with other bachelors, and they're going to look and scavenge around for food. And if they encounter another pride, then they can try to battle, that one male can try to battle another male for control of that pride. And this battling is very, very fierce, and that's why if you see a lot of adult male lions, they have scars all over themselves from these bouts with other male lions. If they're successful, they will then take over that pride, and in fact they'll even kill all other younger males of that previous male because they don't want to raise any cubs that are not directly related to them. So in that sense, Lion King is actually ecologically fairly accurate in that sense. So this idea of being able to move really all has to do with looking for a new pride, i.e. looking for more females in order to be able to reproduce. The next reason why motility has evolved, I think, is also fairly intuitive, and that is escaping from predators, escaping from danger. So prey animals have to be pretty much as fast or faster than predators because they have to be able to escape. So I had to use some really awesome examples here. There are so many that you can choose from, but I chose two examples that I think are fairly unique. And this first one is the Atlantic flying fish. So these fish actually have adapted fins. So they actually have fins in the front of their body and some fins at the back of their body that have been adapted into gliders, or almost like these wings. So what happens is that these fish, like other fish, spend all their time in the water, but when they're being chased by a predator, they can actually beat their tail to provide thrust, and they're able to scoot out of the water, and then by extending out their fins, they can glide over the surface. And if they catch good winds, they can glide over 100 meters. So it's kind of funny, because imagine you're a predator and you're about to catch this fish, and then suddenly it just teleports out of the ocean. It's kind of amazing. And so this is a really cool adaptation that allows them to actually glide over the water and then re-emerge down potentially much further away, and their underwater predator couldn't even see them. So it should be noted though that there are also birds that are aware of this, so the moment that they come out of the water, things like frigate birds are also going to be able to potentially prey upon them. So they can't stay above too long, but they can stay and they can get out of the water long enough to re-enter further away and confuse their predator. So it's a really great strategy. Now, an amazing example of escaping danger has to be the common basilisk. And its ability to run across the water. That's why it's sometimes referred to as the Jesus Christ lizard. And so this is kind of amazing. If you look at their feet, they are frilled and they can actually increase the surface area. And what they're doing is, let's say a snake or another predator is coming nearby and they're going to try to attack this lizard. They can actually escape by running across the water. And the way that they do this is they take their back legs and they basically do this pedaling motion. And because of the high surface area, they're actually able to push down, creating a pocket of air. And because of the surface tension of the water, they don't fall through. And so by doing this pedaling motion, they can run across the water and escape danger. It's really remarkable. Again, think about how confused their predator would be that this lizard just ran across the water. They would just be like it's kind of funny to think about. And the last reason for locomotion is one that a lot of different animals around the world engage in, and it's something called migration. So Part of a lot of animals' life cycle is that they have to physically displace themselves from one part of the world to another part of the world, and that typically is weather dependent. So oftentimes, especially in North America, animals will migrate from a northern area to a southern area during the winter times to try to escape the cold, and then they'll re-emerge back into the north for breeding. This is a generalization, but a lot of migration patterns work the same way. Migration really all has to do with looking for more favorable conditions, and that's really what's going on here. And migration is seen across pretty much so many examples in the animal kingdom. So I want to give you an example of a mammal, of an insect, and as a fish, but keep in mind again that there are many different examples. It's probably funny that I didn't even use an example of a bird here, but you could also use the example of a hummingbird that is also a migratory animal. So probably one of the most famous and the largest mammal migration on the planet is that of the blue wildebeest. It's sometimes referred to as the Great Migration. So these wildebeest travel in herds of over 1 million. In fact, some of the herds have been counted as over 1.5 million individuals, and they engage in this circular migration across Africa that is over 1,600 kilometers long. And really what they're doing is they're basically trying to look for water, and they're trying to look for grass. And so as a herd, they're really well adapted for very long distance, constant movement. As you can imagine, they're actually quite fast in being able to escape predators, and they can actually use herd behavior to try to deter predators from coming in. And if you're wondering, man, if this huge herd is constantly on the move, when do the females have time to give birth? Well, this is kind of amazing. The calf, newborn wildebeest, are actually able to start walking as soon as 2 minutes after being born. So these animals are really like a poster child for they are adapted to move most of their lives. So they're a great example of migration. Now, another example close to home for me is that of the monarch butterfly. And monarch butterflies engage in probably, for me, the coolest migration in the world. And that is because it is a huge distance, multi-generational migration. So monarchs will actually migrate from as high as northern United States, southern Canada, all the way to Mexico and back. Now they do this actually over the course of around 3 to 4 generations. So in Mexico is where the butterflies will be during the winter months, and then they will start to migrate north right around March or so. And as they start to migrate up, a population is going to migrate north, and then they'll breed, and then the new generation will continue to migrate north, they'll breed, third generation will migrate north, and they'll finally hit their northern areas. And then this super generation, this fourth generation, is actually able in one shot to migrate from Canada all the way back down to Mexico. It's really remarkable. So as you can imagine, they have enormous adaptations to be able to fly for such long distances fairly non-stop. It really cannot be overstated how amazing this migration is. And another migration that I want to discuss is a fairly famous one, but the reason I chose this one is that it's really, really cool in that these are fish that spend a portion of their lives in freshwater, but also saltwater. And that is quite rare in the animal kingdom. So salmon are actually spawned in freshwater, right around streams and rivers. And they'll spend a few years there getting larger and larger. And then at some point, they're going to migrate out into the ocean. And that's where they're going to spend most of their adult lives. They're going to get larger there, they're going to feed there. And then when it's time for reproduction, they will then migrate back into the freshwater system, and not just back to any freshwater system, they'll migrate back to the same place in which they were spawned. And then they'll engage in reproduction there. Now, unlike other species of fish that do this, they don't necessarily all die, or they're not necessarily semiparous, meaning that they die after a reproductive event. Some adults can actually survive and then go back to the ocean again to then repeat it later in their life. So they have this kind of amazing migration throughout their life, which is freshwater, get larger, go to ocean, spend most of their time there, then go back to the original freshwater streams to breed again. So they're doing this back and forth between freshwater and saltwater. And that is actually quite unique, again, like I said, with fish. So overall, you should be familiar with at least one example to help explain why motile organisms have evolved to be able to move, i.e. foraging for food, searching for a mate, escaping from danger, and migration. I encourage you to choose an example that is near and dear to your heart and that is easy for you to discuss. And I encourage you to look for other examples other than the ones that I've given. And if you'd like, please feel free to share these examples in the comments. This next learning outcome is specifically about how our marine mammals adapted to swimming. Now, why do marine mammals get their own whole learning outcome? And I think it's because I think a lot of people forget that we have mammals that spend their whole lives in oceans. And you might be like, yes, okay, I know that, but you may not have considered how that's possible, right? Dolphins, orcas, blue whales, all of these mammals, like us, have lungs, and yet they spend their entire time in oceans. That means that, like us, they can drown. How exactly are they breathing? What's going on? What kind of adaptations do they have? Mammals originally evolved on land, and yet marine mammals went back, so to speak, into the ocean. So that means that there must be all kinds of adaptations that have allowed them to make this evolutionary change, so to speak. So let's talk about some of the adaptations that they have. Firstly, let's look at physically what adaptations that they have that allow them to swim very well. And marine mammals can swim at enormously fast speeds, and they're really, really well adapted to swimming, which is again pretty remarkable considering the fact that mammals evolved originally on land. So what kind of adaptations do they have? Well, like a lot of fish, they have this tapered, almost torpedo-like shape that is very hydrodynamic, so that reduces drag into the front. Secondly, they have their flippers on the sides, and this is what allows them to steer. Their dorsal fin at the back, dorsal means back, this allows them effectively to stay upwards and not roll over, so to speak. And then finally, their tail here, or fluke, is actually fused together in this flat part. That's also a really fast way of identifying a mammal versus a fish. The caudal fin on fish are going to be up and down, versus in mammals they're going to be flat this way in a fluke. And unlike mammals, their body is entirely smooth, and they've lost their outer part of their ear, again to minimize drag. So how exactly do dolphins swim? They're going to use their fluke as almost this up and down motion. And anyone here that's done any competitive swimming, as soon as you do a flip turn, you're going to do a very similar motion underwater. The idea is that by actually moving the fluke up and down into this motion, that's what's providing thrust. And again, that dorsal fin is providing stability, preventing them from rolling side to side. And as we've discussed also in B1.1 and A1.1, as a marine mammal, they also have a significant portion of blubber, and this increases their buoyancy, allowing, again, even more efficient swimming. And then finally, an adaptation that you may not have ever considered, like I said, is that these are mammals that spend their time in water, so they have lungs. Now, if their system was like ours, our mouth is connected to our lungs, so that would mean that every time they open their mouth to feed underwater, they're going to drown. So as you can imagine, this system looks quite different. Their trachea actually does not lead back into their mouth. It actually leads to this top structure called a blowhole. And if you've ever had the privilege of seeing marine mammals in real life, or maybe now in video, is that you'll see that when they're underwater, they're effectively holding their breath. They will then seal this blowhole shut, allowing them to dive for potentially many minutes, potentially over an hour. And then when they've run out of air, so to speak, they're going to surface, they're going to exhale, and that's when you see that jet of water come out. Then they're going to inhale and then go underwater again. So their airway has become completely separate from their mouth, and by having a blowhole at the top, it allows them to surface without the risk really of drowning. So it's really remarkable how this system, again, originally evolved for living on land, but now has become really efficient for living in the water. The first part is going to be, what is the anatomy of skeletal muscles? We're then going to look into what is causing skeletal muscles to contract. And then we're going to get into what is actually zooming in, what's happening at the protein level to actually cause muscles to contract. So let's first begin with a little bit of muscle anatomy. Skeletal muscles, as their name implies, are muscles that are attached to bone. The way that muscles are attached to bone are through something called tendons. So tendons attach one muscle to a bone, and they'll attach it not only to the anchor bone, but to the other bone that's going to initiate movement, right? So there's going to be two attachment points, one to one bone and one to the bone that it's moving. If I were to do a cross-section of a skeletal muscle, and that's what this is right here, notice that it's composed of multiple bundles that we call fascicles, and each one of these bundles is then further composed of many, many, many muscle cells. Now you can either use the term muscle cell, that's fine, or you can use the term muscle fiber. These are synonymous. You can use either one. And this is the cell that we've explored already when we did cell specialization. And we talked about that this cell has a number of ways in which it's adapted to its function. Does anyone recall what some of those adaptations were? We are going to be using electrical signals to contract, but by having muscle cells very long, it means that that movement can be spread out over a long distance. So it's a lot easier to do a big amount of movement all at once. We also said that they're quite weird in that they are multinucleated. And the nucleus, as we're going to look here, are actually found on the periphery or on the outside of the cells. And that's really so that they can be packed more of the actual contractile units. And each one of these muscle cells are fed by blood vessels and by a special kind of neuron that specifically signals muscle cells called motor neurons, motor for movement. So again, keep in mind that this is one big cell. You can have muscle cells that are very long and they're all organized. What does this structure remind you of? A rope, a cable, or collagen. What do all of these things have in common? Tensile strength. And in fact, you can even argue that cellulose is organized similarly, where you have a bunch of these almost fibers or cables interwound to provide a lot of strength. So let's now zoom in to a single interior of a muscle fiber or a muscle cell. And that's what this part is right here. So this is just a single portion of one muscle cell. So this is the outside, that would be the nucleus. I'm now I'm cutting into that, and I'm now looking inside. Now inside, you're going to see this extensive network, and this is called the sarcoplasmic reticulum. Now reticulum we've seen already before, and something like a rough or smooth endoplasmic reticulum. Sarco, you're going to see this prefix come up a lot in this topic, and I believe it literally means flesh. So it really refers to muscle. So if you see sarco as a prefix, that's almost always going to be muscle. And if you see myo as a prefix, that's also going to refer to muscle. So the sarcoplasmic reticulum is this network within the muscle cell. We'll talk about what this is later, but effectively, this is a huge storage of calcium. And this is what's going to release calcium to initiate contractions in the first place. Now, it should be unsurprising to you why a skeletal muscle cell has enormous amounts of mitochondria. Why? They're going to be undergoing huge levels of aerobic cellular respiration in order to generate high amounts of ATP, and that will also be more clear later on in this lesson, where you'll see that that process of muscle contraction is very ATP dependent. So they're going to be very mitochondrial dense. And then lastly, what is going on here? Well, you have these bundles of proteins called myo, again, muscle, but not fiber, fibril. We've used this before as well. In fact, when we looked at the structure of cellulose, we said that there were fibers and fibrils. This is another etymology pattern. Fiber, large, fibril, smaller. So remember, muscle cell is muscle fiber. Muscle fibers are composed of many myofibrils within it. This is not a cell. This is within the cell. This is the contractile part. These are the proteins that are actually going to contract together. The last thing here that you may be wondering about is, what is the sarcolemma? Well, again, sarco refers to flesh or muscle, and this is really just the fancy name for the actual membrane of a muscle cell. So we're going to give it a special name. We're going to call it the sarcolemma, but it has the same function as any other plasma membrane. Now, let's take a section of this myofibril and zoom in even more. I'm now zoomed in to a section of a myofibril within a muscle cell. Now, I've color-coded this, and there's effectively 3 major proteins that comprise a myofibril: actin, myosin, and titin. We've already explored actin and myosin when we did mitosis and meiosis, and we said that these proteins were actually used in the contraction of the microfilaments in order to actually separate and organize chromosomes. Actin and myosin go by another colloquial name, and that being this is the thin filament and this is the thick filament. And that's literally because one's a lot thinner than the other. Notice that in the center here, you have these really thick myosin filaments, and they're effectively anchored in place. They're not really going to move side to side laterally. They're going to stay fixed in this position. What's going to end up moving a lot are actin. So actin is bound in these structures called Z-lines, and they expand either way. And notice that there's an overlap between actin and myosin. This is what's going to cause contraction. The myosin and the actin are going to form bridges and actually move over each other in a sliding motion. This is where the name sliding filament theory comes in, is that these filaments, the thin and the thick filaments, are literally going to be sliding over each other during contraction and sliding back during relaxation. And what's holding these parts kind of together is titin. And I think this protein gets often ignored and not really talked about a lot, but it has huge important roles in how all of this comes together. Skeletal muscle fibers and skeletal muscle cells go by another name, striated muscle cells. A striation is a stripe. So if you actually look at actual micrographs of skeletal muscle cells, you're going to see these very clear stripes. So there's going to be a very clear dark band and a very clear light band. Where this occurs is that if you zoom into here, the light band is when there's no overlap between myosin and actin. It's only actin, really. And the dark band is where there's myosin mainly with overlap here. So the myosin is providing the dark part. The light part is only actin. So when muscles end up contracting, there's going to be, the dark band size actually won't change, but the light band will become smaller, and when it relaxes, the light band will become larger. The distance from one Z-line to another Z-line is something called a sarcomere. And again, sarco for muscle, and this is effectively the contractile unit of a muscle. If we were to measure or gauge the degree of contraction, we would basically assess how short is a sarcomere becoming and how long is a sarcomere becoming. Because as a muscle contracts, Z-lines are going to come closer to each other, sarcomere distance decreases. As a muscle relaxes, sarcomere distance is going to increase as the Z-lines move further and further apart. I've already kind of alluded to the fact that the contraction is going to be the relative overlap between actin and myosin. So what is titin doing? Titin is not only acting as an anchor point, holding these all together, but as something we're going to explore in a second, it's also hugely important in allowing relaxation to occur through recoil. So it's going to act almost like a spring throughout this entire process. I think the logical way that I want to explain this is, first, let's talk about how a muscle signal is actually sent and how do we initiate muscle contraction, and then we'll look into what's actually happening at the protein level. When we send an impulse through your nervous system, that is an electrical impulse. All electricity is the flow of a charge. And in the case of our neurons, that charge is going to be sodium and potassium. So we're going to be describing how is a signal sent to cause a skeletal muscle to actually contract. So the neuron involved here is a motor neuron, as I mentioned already. So a motor neuron is going to send an electrical signal that we can call an action potential. And that's going to travel from the soma, or here, dendrite to soma to axon. It's going to flow down very quickly, something like 200 meters a second. And it's going to ultimately end up on these end parts. And the same term that we use for like the end of a line on a subway line, we say it's a terminal. So if this was the axon, these would be axon terminals. So we're going to zoom in to here, something called the neuromuscular junction. Neuro for where is the neuron connecting, so to speak, with the muscle. So neuromuscular junction. So notice though, there's a gap. They're not actually touching. And that's true for all neural pathways. If we're sending a signal from one neuron to another, or one neuron to a gland or a muscle, they're not physically touching each other. Now this might seem counterintuitive, because wouldn't that be slower? Kind of, but it does allow control. It means that we can determine, so to speak, when a signal goes from here to here, and we can modulate that signal. So this gap, this junction between, is something called a synapse, or the gap is referred to as a synaptic cleft. When we look into neural signaling, we'll talk about how an action potential is generated. That's not necessary for this subtopic. All we want to talk about here is how do we get a signal from the motor neuron to the muscle, and how does that ultimately cause muscles to contract? So an action potential is coming down. Now sodium, which is really what an action potential is, it's a huge influx of sodium. Sodium is now going to cause a different voltage gated channel to open. Take a look at this diagram. If sodium is opening voltage gated calcium channels, look at the direction of calcium. What might we infer as to the concentration gradient of calcium? Must be high outside, low inside. So as the voltage gated calcium channels open, calcium is going to diffuse from outside of the neuron into the axon terminal. And calcium is actually going to act as a signaling molecule. In fact, we've already seen calcium's potential as a signaling molecule when we looked at reproduction. Calcium acted as a signal firstly for the release of the acrosomal enzymes, right, in the acrosome reaction, and calcium was also involved in the cortical reaction, where that actually causes the cortical granules to release their enzymes via exocytosis, hardening the zona pellucida, right? So this is also going to be a recurring theme in this course as well, that signaling molecules aren't just proteins or carbohydrates or whatever. They can actually just be ions, and calcium is a great example of, it's a very simple ion, but it acts as a signaling molecule. So in this case, what exactly is calcium signaling? Well, it's signaling this vesicle. So this vesicle is actually containing a bunch of neurotransmitters, specifically one that we've explored when we looked at membrane transport, acetylcholine. So acetylcholine right now is stored in these vesicles. Calcium basically signals these vesicles to move to the periphery and undergo exocytosis. So If it's high here, it's going to diffuse across the synaptic cleft from the motor neuron axon terminal or the presynaptic cell to the postsynaptic cell. In this case, this cell would be the muscle fiber. This is now going to be the protein that we examined in membrane transport, that being the nicotinic acetylcholine ligand-gated channel. So nicotinic in the sense that nicotine as a chemical or a class of chemicals can all bind to this. If this was a voltage-gated channel, meaning it opens and closes relative to the change in membrane potential, this is a ligand-gated channel. It's going to open and close depending on if a ligand or a chemical is bound to it. So acetylcholine is now going to diffuse, bind to this acetylcholine receptor. That causes the acetylcholine receptor to open, allowing an influx of sodium. Now, why would sodium diffuse in? Because a sodium-potassium pump is constantly keeping this gradient. That's always going to be a recurring theme, right? Anytime that we ever talk about membrane transport and ion transport, that sodium-potassium pump is going to be something that we discuss all the time, especially in this overall physiology unit, because we purposely are keeping a high gradient of sodium out, high gradient of potassium in, and that is going to be used a lot to do work for us. So sodium is now coming in. We now get a new action potential that's been generated. This is now going to pass along the sarcolemma, and it's going to pass down a special channel. That's what this is up here, these T tubules. So what happened is that we now got basically sodium is now going bop, 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 and then once it hits a T tubule, it rushes down the T tubule. And then what's it's going to do? It's going to signal the sarcoplasmic reticulum. So this is now going to result in the sarcoplasmic reticulum releasing yet again calcium as a signaling molecule. So now calcium is going to be released from the sarcoplasmic reticulum, and that's what's going to initiate muscle contraction. If acetylcholine is being released here, is this signal just going to happen forever? Well, no. If it did, that would be paralysis. And in fact, there are some drugs and some toxins that can cause that to happen. What's happening here is that something that we'll discuss later, but we actually have enzymes that break down neurotransmitters to then recycle them back. And that causes the signal not to constantly happen unless we want it to. So that's how a signal is sent from a neuron to a muscle. So the end result was a huge release of calcium. That's now going to cause your muscles to contract. We're now going to zoom in to here. Let's do a zoom in on just one of these sections, and we have this right now. So in blue, that's the thin filament, actin. In red is the thick filament, myosin. So notice myosin has these bulbous, huge heads at the end. That's what's going to be able to ultimately bind to actin. Now, when your muscle is relaxed, why can't these two bind? Well, there's something in the way. There's another filament blocking that connection point. So overlaid on actin is another protein called tropomyosin. Tropomyosin is currently preventing or blocking this myosin from binding to actin. It can't. So when your muscle is relaxed, the two physically cannot join. Notice on tropomyosin, there's a receptor. That receptor is called troponin. Notice that troponin has a binding site. It's going to bind what we just released, calcium. So now that calcium was just released from the sarcoplasmic reticulum, it's now going to bind onto troponin. As it binds to troponin, it's literally going to lift the tropomyosin out of the way. This now frees the actin binding sites, and the myosin is now going to be able to form what's called a cross bridge, or a bind between the myosin head and the actin. These two have now formed a bond. Now at the moment, myosin is already bound to ADP and an inorganic phosphate. That's going to become important in a second. So the two are now bound. We're now going to go over the cycle of what's actually occurring as your muscles contract. So we now have formed an actin-myosin cross-bridge. As ADP and inorganic phosphate unbinds, this myosin is literally going to pull almost like a ratchet on actin. This is going to physically move this Z line in. So as the myosin literally does this, the Z lines come in, if that makes sense. So it's almost like a ratcheting motion. Then ATP is going to bind anew to myosin. Once ATP binds, that cross bridge is broken. So it now unbinds. So we've ratcheted it in, ATP binds, myosin, actin, unbind. Well, what do we do with ATP? We hydrolyze it. As we hydrolyze ATP, we've now formed ADP in inorganic phosphate, but what was the point of that? Well, you effectively changed its protein conformation. The verb you can use is that the head is in its cocked position. So it's almost like, the way that I think about this, it's almost like a mousetrap. The way a mousetrap works is that if I'm setting a mousetrap, I'm transferring potential energy into that actual spring, right? And then I'm locking it in place. And then the moment I release that, all that potential energy that's been stored can then swing forward. So this is the analogy that I have to try to help wrap my head around this. So hydrolyzing ATP stored potential energy in the myosin by cocking the head back. Then, what do you think is going to happen? We're going to restart the cycle again. This thing then binds and then ratchets strap forward. So we get this cycle happening over and over and over again. So we have myosin-actin cross-bridge, power stroke, moves the Z lines closer to the middle, or the M line. ATP binds, breaking the cross-bridge. ATP hydrolysis, cocking myosin head back. New cross-bridge formation, power stroke, over and over and over again. So what you end up getting is you get this Z line to Z line movement closer and closer and closer. So if you were to compare your sarcomere during relaxation, look at the relative size of the I band. As the Z lines become closer and closer and closer, there's more and more overlap between the actin and the myosin, shortening the I band. So you start to see that the actin and myosin are sliding over each other. So when your muscle is literally contracted, how did it get shorter? Well, because it got taller, right? That you started out like this, and then you moved it close together, bulging your muscle out, but shortening it overall. So that's what's happening here. The two filaments are literally sliding past each other to cause a shortening of that muscle. When you're describing this motion, you don't want to say it's just moving the Z line. It's moving the Z line where? You can either say it's moving the Z lines closer to each other, that's fine. Or if you want to use, that's the only reason I included M line here, is it's like a identification point. You can describe this motion as Z lines coming closer to the M line, or the middle line, really, is what's going on here. Now, in this whole process, you may be wondering, well, what's the point of Titin? And honestly, Titin is kind of forgotten about a lot, which is kind of odd because it is composed of, I think, over 30,000 amino acids. It is composed of hundreds of thousands of atoms, a single molecule. It's huge. It's an enormous protein. So, yeah, hence the name. What exactly does Titin do? Well, take a look at the relative appearance of Titin when the muscle was relaxed and when the muscle is contracted. When the muscle is contracted, Titin is actually compressed together. And when the muscle is relaxed, it is stretched. If I stretch something, then I'm actually increasing elastic potential energy. So as a muscle is relaxing, titin is becoming stretched, storing potential energy. That means that when its antagonistic muscle is contracting, the two are working in tandem. When one muscle is contracting, titin is coming compressed, but in its antagonist is getting relaxed. And so when the antagonist flips as now being the agonist, titin can now come back, releasing its potential energy, allowing relaxation to occur. I think the misconception about titin is that I think a lot of people assume that springs are only adding potential energy when you compress them. That's one way. But also keep in mind that when you stretch something, you're also adding potential energy, right? Because when you stretch a coil and then release it, it springs back together. And that's really what's happening to titin. So that energy that's used to stretch titin, it seems like where is this energy coming from? Because muscles can only exert force during contraction. But because muscles work in antagonistic pairs, titin is actually stretching in the relaxed muscle when its antagonist is contracted, and vice versa. The other role of titin, I think, is fairly intuitive if you look where it is. It has a literal structural role. It prevents overstretching, right? We don't want your sarcomere to become too stretched. You can actually cause damage. So titin is actually almost like an anchor, preventing overstretching. Now, this whole process eventually will stop once calcium is either depleted or is no longer being released. So if you look at this now in the reverse direction, calcium is then going to leave, tropomyosin goes back into position, and myosin and actin can no longer form a cross bridge. And hence, that's why in its relaxed state, it already has ADP bound to it. Hopefully, after looking at this whole process, you start to now really appreciate why movement is so ATP intensive. Every single one of these ratcheting motions at every single one of these myosin heads took ATP. So that's multiple ATP per myosin times how many there are in a muscle just for a contraction. So it's massively ATP dependent. Hence why there was so much mitochondria within a single muscle fiber. So now that we understand how muscles actually contract, let's finally look at how muscles actually cause movement in organisms. And I think the best place to start with that is looking at anatomy. So I think when we hear the term skeletal muscle and you think skeleton, what you're probably thinking of is an endoskeleton, meaning a skeleton inside of the body. So that's the case with vertebrate animals. So things like mammals, birds, reptiles, amphibians, bony fish. These organisms have their skeleton inside their body, hence an endoskeleton. And if you were to look at anatomy of this, what that would look like, and let's say the arm, would be that the bone is inside of the actual limb in this case, and the muscles being attached to the bone right here. And so this would be an example of an endoskeleton where a contraction of this muscle would lift the arm and a contraction of this muscle would extend the arm. However, a type of skeleton that you may not have considered is that of an exoskeleton. Exo for outside. So in this case, the skeleton in these organisms are actually on the outside of the body. So it may look a little odd, but let's say I'm looking at the limb of, let's say, an insect, like a grasshopper. The outside part, this is the actual skeleton, and the muscles would actually attach from the inside portion. As these muscles contract, it actually causes the leg to either flex or other extend. So what kind of organisms have an exoskeleton? So these would largely be arthropods or invertebrates, things like insects, arachnids, crustaceans, myriapods. These organisms, their skeleton is found on the outside of their body. This comes with some extra problems, and for instance, consider what would happen as an organism is growing with an exoskeleton. If an organism is getting larger and larger and larger, the exoskeleton can't really expand. So what's actually going on here? So in this case, these organisms would actually do what's called molting, where they would shed their exoskeleton, physically allow themselves to get larger, and then they would rebuild their exoskeleton again. So now's probably a good time for me to talk about what are the exoskeleton actually composed of. Well, in many arthropods, it's actually composed of a material called chitin, such as in the case of a lot of insects. But this is not true for all organisms. So another major building block of exoskeletons would be things like calcium carbonate, as in the case of like shells and corals. So both of these materials are very hard, strong materials, and this is what's actually on the outside of the body. So in either case, both an exoskeleton and an endoskeleton, the skeleton is providing the actual anchoring point, and the muscles are the ones providing the power. To understand how muscles really work, I think we have to consider joints and how joints are composed. So in a simple lever system, you have a few major components. One would be the fulcrum, which is, this is the actual pivot point. The lever is that rigid beam that moves over the fulcrum, and then you would need some kind of effort or force applied that's actually going to alter that lever, and of course the load, what you're actually moving. If you take this simple physics concept, we can apply this to standard joints. So consider how our arm would follow the same point. The fulcrum would be the joint, in this case the elbow. The bones are going to act as the strong structural levers. The muscle is the one that's going to be providing the force, and the load is the physical bone and maybe any weight that you're actually using to lift this here. Of course, the larger the load becomes, the harder it is to actually move this joint. But all of your joints have this same principle: a fulcrum, a lever, a force, and a load, and the muscles are the ones providing the force. Now, it is worth noting a little bit about biomechanics. There are many different types of levers. We're not going to be dealing that with in this course. So consider this example where the limb is actually relatively long. So this kind of setup would be quite useful as it allows a larger range of motion and a greater acceleration over a distance. So you see this a lot in things like short bursts of power or things like sprinting. Hence why something like a cheetah would have very long legs and why this is really efficient for running. However, if you're going to try to do a lot of power, a long limb is actually less efficient. So the opposite is also true. If the limb is actually very short, this allows, while less range of motion, a lot more power. So something that, like, let's say, very short arms in moles, allowing them to dig and provide a huge amount of power, is really, really useful here. So biomechanics is actually a really interesting study in that it looks at the actual efficiency of certain levers and certain limbs and how these all work together. I mean, I think we already intuitively know this concept. Think about how much more difficult it is to lift a weight just using your arms versus lifting your entire body weight on your tiptoes. These are two different kinds of levers, but they're organized in much different ways, allowing different efficiencies to occur. Now, something that we've explored a lot in this course already is the idea that muscles work antagonistically. A quick way of remembering this is that muscles can't actually push, they can only pull. Let's imagine a door is what I'm trying to open and close, and the only way I can do so is by attaching a rope to the handle. I can pull on that door, causing the door to actually swing in this direction, but I can't push a rope. I would have to pull from the opposite direction. So this means that in any type of joint, if I want bones to move, or I want the skeleton to move, I'm going to need two sets of muscles that are going to work antagonistically to each other, meaning when one muscle is contracted, the other muscle is relaxed. So let's look at an example of, let's say, an insect leg, such as in the grasshopper. So here I have an exoskeleton shown in green. The muscles would be inside of the skeleton. So in this case, we have an antagonistic pair, something called an extensor and a flexor, named after the fact that when an extensor is contracted, it extends or lengthens a limb, versus when a flexor is contracted, it bends the limb this way. So in the case of the leg, when the extensor is contracted, this actually pulls the leg up whereas the flexor at this point would be relaxed. But now that the leg is out, if I want to contract it back in, I would actually relax the extensor and pull on the flexor. And notice the attachment points are slightly different, causing this range of motion to occur. So when the extensor is contracted, the flexor is relaxed and vice versa. So these work antagonistically. This would also be true, again, in a very simple example of, let's say, your arm. If I want to pull my arm up, I'm going to flex my flexor, such as my bicep. This is pulling my arm up, because as this shortens, it's almost like, imagine a rope on my hand, literally pulling this up. But if I continue to contract my bicep, this arm is not going to move in that direction. I need something to pull it back in this direction. So that would be contracting now its antagonist, which would be the extensor, which is my tricep. As I contract this, it's almost like a rope pulling my arm back down this way. So the bicep and the tricep are working antagonistically to allow movement across a joint. Now, the main example that we're using in this topic is actually one that we've already explored in B3.1, which is ventilation. So the muscles in between your ribs, your intercostal muscles, remember that means inter or between ribs, or bone. In this case, the intercostals, you have two different kinds, recall. You have the external intercostals and the internal intercostals. And so during ventilation, you want your rib cage is going to expand out during inhalation, and during exhalation, it's going to compress in. And your internal and your external intercostals work antagonistically in order to accomplish this. So if I'm doing inhalation, then the external intercostals are actually going to contract, and as they do that, they're lifting my ribs up and out, and again, resulting in an increase in thoracic volume. But while that's happening, it means that the internal intercostals are actually relaxed. Now, if my ribs are up and out, and I now want to compress them again, then the exact reverse situation is going to occur. So now, when the internal intercostals contract, they literally pull my ribs back down and in, and the external intercostals would currently be relaxed. Now, the reason that this works is, notice that the internal and external intercostals orientation is different. As the externals are oriented in this direction, when they shorten, they will literally pull the rib up. But as the internal intercostals contract, they will literally pull the ribs down. Now, the next part to describe in this same process is one that we've just previously discussed, which is, what is the role in Titin in all of this? Titin, recall, is able to store potential energy when it stretches. So let's say this is the sarcomere within the external intercostal, and this is a sarcomere within its antagonist, which would be the internal intercostal. Now, when the external intercostal is contracted, notice the Z lines are close together, and Titin is currently compressed. At the same time, this means that in the internal intercostals, because they're relaxed, this is stretching this out, and as Titin is stretching, it's storing potential energy. This means that when now I would be exhaling, this is going to recoil back and allow this sarcomere to spring back easier, and this would then stretch out in the external intercostals. So in this way, because your muscles are working antagonistically, when one is contracting, it's allowing potential energy to be stored in the stretching of Titin in the other muscle. The role of Titin is really, really obvious when we look at the interplay between antagonistic muscle pairs. So we have two bones here. Now, coating the tips of the bone is going to be cartilage. In between both of the bones is going to be the namesake of this type of joint. A synovial joint is going to be synovial fluid. Now, containing the synovial fluid within this joint, so it doesn't go anywhere, is something called the joint capsule. So we have two bones coated with smooth cartilage, and in between both of them is a cavity that has synovial fluid being contained by a joint capsule. Now, these two bones are not just free floating, they're actually physically attached to each other through something called a ligament. And finally, what's actually providing the force here, and that would be your muscles. Now, if a muscle was just contracting like this, it would cause no movement. So what's actually causing it to move a bone? Well, it's connected to a bone via a tendon. So overall, these would be all the major components of a simplified synovial joint. Now we're going to explore this more specifically in a named example, that being a hip joint. Now the hip joint is a special example of a joint, and that being a ball and socket joint. And what that means is one bone is actually going to have a rounded end, that's the ball, and another bone is going to have almost like a cavity or cup, and that is the socket. So the ball is going to be able to rotate around in that socket. This type of joint enables the greatest range of motion. This allows for extension and flexion, moving my leg back and forth in this direction. You also have abduction and adduction, meaning I can move my leg back and forth in this way. And finally, you even have internal and external rotation. So there's a huge degree of movement in a ball and socket joint. So what does this actually look like in the hip? So here is some anatomy of the hip. In front here is a straight on view of the hip, and I showed on this diagram the bones, the tendons, the muscles, the ligaments. So you can see how these are all intermingled here. So the socket part is going to be the pelvis, and that's where this cusp is going to be. And the ball part of the joint is going to be your femur. So if we look here, this femur part has the ball like this, and the socket is the pelvis right here. So this is where that rotation is happening. Now, of course, we have a lot of different muscles involved here, and it's not necessary for you to be able to name these, but these muscles, again, would be attached to both the pelvis and the femur via tendons, and the bones would be attached to each other via ligaments. Now here is a zoom in or a cross section of this bone. So let's imagine I'm now doing a cross section this way, and I'm looking into this joint in this direction. Here you can now clearly see the femur acting as the ball. Here would be the socket, which would be the pelvis. We have the ligaments holding these bones together with each other. And then in blue, here is the synovial fluid. On either end, lining the pelvis and lining the femur is cartilage, and we have the joint capsules holding that all in together. So what is the role of all of these parts? So the bones provide anchorage. So the femur as the ball and the pelvis as the socket are each providing anchorage around this joint. The cartilage, the reason why we're lining the tips of the bones here is that cartilage is very smooth and actually slightly rubbery, so it actually not only can absorb shock, but it massively reduces friction. And if we want a joint to be very, very efficient, we would try to reduce as much friction as possible. The synovial fluid, again, if you've ever had a squeaky door, what are you going to do? You're going to add oil. If you ever had a squeaky bike when you're trying to shift gears, what are you going to do? You're going to use oil. Oil is acting as a lubricant, meaning it is reducing friction by providing a layer between moving parts. The synovial fluid is doing the exact same thing. The two bones are not actually rubbing together. That would be incredibly painful and inefficient. Instead, they're actually moving in a slight space that's covered by this fluid. And again, the joint capsule is the physical tough coating that's preventing that fluid from leaving the joint itself. The ligaments would be the connective tissue physically holding two bones together, and the tendon is what's actually holding a muscle to the bone. And lastly, of course, the muscles, that's actually what's providing the force of movement. So this learning outcome here is, for one, given a diagram, you should be able to label the different parts of the different anatomical components of this joint, but also you should understand how these parts are adapted to their function. How do these things actually enable a smooth motion? Now, it should be noted that not all of your joints, of course, are ball and socket joints. Some of them actually restrict movement in a certain way, and that would be your hinge joint. Great examples here would be your elbow or your knee. They only allow movement actually in one plane, which would be extension and flexion. And so in this case, their orientation is going to be slightly different, but these are also synovial joints. And understanding how different joints all work together is, again, the study of biomechanics and kinesiology. So if you really are interested in that, I would recommend looking into these areas of study. Now, the last part here is actually being able to measure the degree or range of motion along a joint. Now, this might seem, what is the purpose of this? Well, like any other measure, it's useful for a doctor and yourself to know what a normal range of motion for yourself would be and if this range of motion is being impaired. So let's say I go to my doctor and I'm saying my shoulder really hurts, I can't seem to move it as much. Well, being able to know what my standard or normal range of motion should be and then what my current range of motion is would allow the doctor to now understand how impaired this joint is. And throughout rehabilitation, they can then gauge how am I improving or not improving? Is my range of motion increasing or decreasing? And to do that, we're going to use something called a goniometer. And so a goniometer is actually quite simple. I actually created this from this website, just printing off it and gluing it on some hard stock. And then I just used a little pin here to link these together. So you would line this up over a joint, and then you would ask the patient to move their joint, and keeping this part, the bottom limb, steady, the upper limb is going to move with the person. As they do their normal range of motion, this gets removed, and then you can literally follow the arrow to see how many degrees or range of motion would that be. And for every single joint, there is a normal range of motion that's stated, but it's also useful to know your own range of motion, again, to see if you're improving or getting worse in that range of motion. And this could be a sign of potential injury. Now, this is a manual way of measuring this, but there are also digital methods where you could actually, let's say, photograph my arm and photograph it again, or using computer analysis, you can determine the actual range of motion that just occurred. So overall, this whole section was really all about how do muscles allow movement in a joint, and what are the different components of a synovial joint, and how can these all ultimately be measured using goniometry. Okay, let's look at the learning outcomes for subtopic B3.3 Muscle and Motility. The very first learning outcome here is we explored the concept of just movement and how that's a feature of all living organisms. But we explored how this might look different in motile and sessile species using a number of different animal and plants as key examples. This subtopic explored the actual sliding filament model of muscle contraction. And we actually explored how the sarcomere, which is the contractile unit of muscles, actually contracts by sliding actin and myosin filaments over each other. We explored the often forgotten protein of Titin and its role in the antagonistic muscles during contraction and relaxation. This learning outcome was exploring what the actual anatomy looks like when the nervous system interacts with the muscular system, and that of a neuromuscular junction, and what the role of motor neurons and muscle fibers are and how they interact with each other. This learning outcome explores the roles of skeletons as anchorage for muscles and how they act as levers in a simple system, including a fulcrum and the force and the load, and how they actually work with respect to biomechanics. And we also explored that skeletons are not only endoskeletons like invertebrates, but also exoskeletons in arthropods. This was further explored by looking at how movement actually occurs at a synovial joint. And students should understand the different roles of these different structures using the human hip joint as the key example. And then finally, with respect to joints, we looked at how the range of motion can really differ and how this can be measured using a goniometer. Going back again to antagonistic muscle movement is we explored how the internal and external intercostals really work well because they work antagonistically, and how this actually helps facilitate movement, and in this case, ventilation. This learning outcome explored what the actual need for locomotion is. Not all organisms are able to do locomotion, so those that can, there must be an evolutionary advantage. And that included foraging for food, escaping from danger, searching for a mate, and migration. And we explored a number of examples of each of these. And then finally, we explored how different marine mammals are adapted to swimming. And this is of particular importance because mammals originally evolved on land, and we explored how the different structural features have been adapted over time to allow them to be so successful in the water. If you want to learn more, which I always encourage you do, this back page is a really good place to start. Right here are the different textbooks that I used in order to develop the content for this lesson and the relevant page numbers. These simulators are really, really useful in trying to actually practice our understanding of muscle contraction and actually being able to visualize it really, really well. These 3D models explore the key proteins involved in the sliding filament model, that being actin, myosin, and titin. And these different articles from the PDB 101 really help explore this structure more in detail. This right here is an interactive of the human hip joint, which I encourage you to take a look at as you're able to manipulate it more in 3D space and you're able to get a better understanding as to how these work with each other. And then finally, I find these articles really, really interesting as a further reading on this topic. This one was exploring titin and how it's often the forgotten protein involved in muscle contraction and why it's so important. This one is a really nice summary of goniometry and how this can be used to measure various joints. This article explores the hip joint in detail. And finally, this is a really nice paper kind of summarizing our understanding of the sliding filament model of muscle contraction. Finally, I would always encourage students not to learn for the sake of a test, but learn for the sake of learning. Just learn for enjoyment.